Bismillahirrahmanirrahim. Thank you very much for inviting me to participate in this conference. I am grateful to Dr. Asif for allow me to present my presentation in this event. Metagenomics is an important aspect of biotechnology these days in modern molecular biology and genetics. Metagenomics can be applied metagenomics can define as the study of genetic material obtained directly from the environmental samples, including soil, water, and human body. Virtually any sample can be used for metagenomic analysis, which contains DNA or RNA. Metagenomics uses next generation DNA sequencing technologies to analyze DNA of all the microorganisms present in a sample, providing insight into the diversity and functions of microbial communities. Metagenomics has a wide range of applications across various fields, including medicine, environmental sciences, agriculture, bioremediation, in food industry, in biotechnology, in drug discovery, and many more areas where metagenomics can be applied. Metagenomics can be carried out by two methods, two approaches. First is called as shotgun metagenomics, and the second is called as marker gene based metagenomics, or commonly termed as 16S ribosomal DNA metagenomics. This slide is showing that is how metagenomics is carried out. So first, basically, we collect DNA, extract and purify DNA from any sample, and then after that, the ribosomal 16S ribosomal RNA gene is amplified, and from all of the DNA sample, and then this 16S is then sequenced by using next generation DNA sequencing machines, and after the sequence is obtained, the sequence is then analyzed using an array of bioinformatics software, and then which result in the taxonomic classification of sequences present in the sample. And also we carry out community composition analysis and phylogenetic analysis of the DNA sample present in the environmental sample. And after that, finally, diversity and statistical analysis also can be carried out. Mostly nowadays we use 16S ribosomal RNA gene for phylogenetic analysis present of microorganisms present in any sample. Having said that, now I would like to mention some of our experiments and research we have carried out using metagenomic approaches. We applied metagenomics approach in biomedicine, in the analysis of the human microbiome. We know that human microbiome is a very important aspect, and there are microbial cells which colonize our body, and they are at least as abundant as our somatic cells in our body. So there are trillions of microbial cells in every individual's body. They are colonizing in our body, and so the microbe contains more genes than the human genome, because there are thousands of hundreds of different species of bacterial species are present in human body at any time, and this collectively called as human microbiome, which offers substantially more genetic diversity and flexibility than the human genome. Human harbors diverse microbiota, including bacteria, viruses, fungi, protozoa, and archaea. So different classes of microorganisms are there in our body, especially in particularly bacteria. And human bacterial flora is essential for maintaining health, as we know. And there are different body sites where human microbiome is present, including oral cavity, our gastrointestinal tract, and other places. There are many factors which influence human microbiome, body site, for example, our genetics. Immune reactions also affect the that is the amount of and diversity of microbiome. Our diet also is very important in affecting human microbiome, lifestyle, the antibiotics we take, and there are other factors which affect human microbiome. Human microbiome is called as highly personalized. It is not specific. It is not that is generalized. It is very specific, and it is highly personalized and highly dynamic also. So at different time, different condition, different age, our microbiome is changed. So I will present my work on related to our oral bacterial flora analysis. So there are hundreds of bacterial species present in oral cavity, in our oral cavity, in our mouth. And these are different phylogenetic representatives of bacterial species. And it is said that in different conditions, in different that is disease conditions, the oral bacterial flora has been observed to be changed. Salivary bacteria are also has also suggested as biomarkers in cancer. And it is also, there are reports that salivary bacteria has been associated with diabetic conditions. Okay, there are several reports I have mentioned over here. I'm not going into the detail because of lack of time. So what I want to present over here is the bacterial communities in oral cavity of type 2 diabetic patients, the study which we carried out in Karachi. So basically we collected saliva of normal individuals and diabetic individuals, and then we collected their DNA from saliva and then perform a metagenomic analysis using next generation DNA sequencing techniques. And after that, we carried out a comprehensive bioinformatics analysis of NGS data of the metagenomic DNA using an array of bioinformatics software. Okay, so these are basically the PCR amplification images, which is one of the techniques involved in metagenomics analysis. And after that, the sequences were carried out, and then after bioinformatics analysis, we found that some specific phylogenetic data, some specific type of phyla, bacterial phyla, are predominantly present in the saliva of diabetic individual compared to the healthy individual. For example, we noted that in diabetics, proteobacteria are decreased, statistically conserved, and some other bacteria are also decreased or increased in compared to the control. As far as our phylogenetic analysis, the bacterial phyla analysis, we basically carried out a statistical analysis using several statistical software, and we found out that proteobacteria are decreased in diabetic individual statistically significantly, and firmicutes are increased in diabetes individual, and bacterotides are also increased. So there are indication of that is imbalance of normal bacterial flora in diabetic individual oral cavity. This slide is showing us the analysis of differential abundance profile of bacterial genera. That is the analysis at general level, the genera genus level, which is and compare as because phyla is a very big that is collection of bacteria. In a phylum there are hundreds of different families and species, but so we carried out this analysis based on the bacterial genera, and we found out that in diabetic individual, Prevotella genus related to bacteria related to Prevotella genus are increased compared to normal individual. Likewise, Veillonella genus bacteria are also increased in diabetic individual compared to healthy individuals. With the more than 95% confidence interval. So and some bacteria are decreased in diabetic individuals, including Pseudomonas, Sprentophomonas, and Porphyromonas. So we were we thought that what is the consequence of what might be the consequence of this increase of some bacteria in diabetic individuals oral cavity and decrease of some individual of diabetic individual. This slide is showing us the gender-based heat map plot of top 40 bacterial genera, comparison of male and female diabetics and healthy individual. And this slide is also providing statistical analysis of abundance of differential abundance of bacterial genera in healthy male and females compared to diabetic male and female. So this data showed us that there are different bacterial abundance profile in male and female. There are many reasons related to that. So in conclusion, we, as I mentioned, that at genus level, acidogenic bacteria, Prevotella and Veillonella, are that is increased in diabetic individuals. They basically produce this type of bacteria produce at because they are so-called acidogenic bacteria. So they produce organic acid after fermentation of sugar, and this organic acid then that is involved in dental biofilm degradation and other periodontal diseases in mouth. So this is our observation and our finding. So we have to further confirm it with other data, and we are doing more research in this and carry out further investigations. The second study which I want to very briefly discuss about the metagenomics analysis of salivary bacteria in the addicts of betel nut preparation. As you know that in Karachi there are the betel nut preparation, that is chalia and supari, and this type of pan and pan masala and this type of things are being used in a specific population in Karachi and other parts of Pakistan, especially in Sindh region. So we carried out betel nut, that is the salivary analysis of those individuals. Those are addicts of betel nut preparation, and we call this as the gutka chewers and non-chewers. And we found out that there are considerable variation in bacterial flora of the people who are using gutka and related betel nut preparation. And we found out this slide is telling us about the PCA principal component analysis graph is showing that the gutka chewers microbial oral microbiome is considerably different from the oral microbiome of non-chewers. So they are the bacteria which is present in are much more different, and that is their bacterial community are different compared to the non-chewers. And we have seen that in gutka chewers, as Serratia, Valonella, and Streptococcus genera are more in abundant compared to other compared to non-chewers. Our third that is um, study was on the bacterial community analysis in drinking water supply system of Karachi city using metagenomics. The paper was published in Microbial Ecology, and it is very much cited from many people around the world. It was the first study of the metagenomic analysis of the drinking water which is being supplied in Karachi. We collected water samples from 20 different places and from the sources of Karachi water being supplied in Karachi, that is the Kinjar Lake and Hub Dam. And then we carried out in metagenomic sequences followed by bioinformatics analysis, and we found that there are considerable differences in the bacterial microbiome in the water samples being supplied in Karachi. Okay, and there are different that is analysis, observations we found out, and we reported this in our paper. Recently, the similar study was published in our collaboration from Peshawar, in which the drinking water system was used for metagenomic analysis, and the paper was published in Water with our collaborator in Khyber Medical University. The second last study which I want to mention over here is the metagenomic profiling of freshwater lakes at different altitude in Pakistan. It was a very interesting study in which we collected water and performed metagenomic analysis from Lake Saiful Malook in Kaghan region, and from Rawal Dam near Islamabad, and Kinjar Lake near Karachi. And then we carried out their metagenomic bacterial genomic analysis, and that is their gene annotation, what type of genes are there in these bacteria in these waters in these lakes. And our highlight of this study was the antibiotic resistance genes, which is present in the water sample of these lakes. And we found out, interestingly, that the water present in Lake Saiful Malook is containing bacteria which contains higher number of antibiotic resistance genes. The last study which I want to mention over here is the metagenomic analysis of the mud volcano in Balochistan, in Lasbela district near Karachi in Balochistan. We visited over there, collected sample from different mud volcano. Is a very interesting geographical and geophysics phenomena which is present and very unique in Pakistan, and we carried out the metagenomic analysis, the mud volcano, and we found out that more than 90% bacteria present in this mud are Serratia. It's simple, it's not very complex microbiome over here. So, having said that, because of the time constraint, I would like to mention my collaborators who have been working with me in metagenomic analysis and microbiome studies. My student Junaid is in Jamshoro. Faizan is currently in Toronto doing his PhD. Ghulam Mushtuba is doing MPhil. We have been collaborating with Dr. Atif Atif Mustafa in NED University, Dr. Zahra Hassan in Aga Khan University, Dr. Shahab Abid in Aga Khan University, Umar Baisal in Turkey, and Mariam Shafiq in Federal Urdu University. And finally, I invite all of the participants and to submit their paper in our journal of Pakistan Journal of Biochemistry and Molecular Biology, which can be accessed by pjbmb.com. Thank you very much for your kind attention.